Okay, good afternoon everybody. So as you can see, the particles have been moving through the different work packages and now they've reached the first in vivo stage. So in this part, we have data concerning an objective, which was to examine the fate of these peptide-loaded nanoparticles in rodent models. So really, the education piece here is all about seeing whether the methodology we used is going to be useful to both track the particles and also to see if they're going to cause any local immunology. So we've got some fluorescent probes that are attached to the nanoparticles themselves, and in some cases, fluorescent probes that are attached to the peptide carrier. Okay, and then the second aim then was to see if there's any local gut immunity caused by these nanocarriers in mouse models. So we have some positive data in an installation model in rats. So this is under anesthesia. It's isolated loops of intestine in the anesthetized rats. So they have a blood supply. We're measuring peptide coming through into the blood, and we're also measuring pharmacodynamic effects. And in this example, from one of the nanocarriers that we had in the consortium, we've got an effect of lowering the blood glucose in this example with the peptide in the nanocarrier. And we also, in parallel, we have insulin levels actually going up, which is measured by ELISA. So we've done this a few times with different carriers, and we've gone through about 7 or 8 of them, and we've got effects of some of them, which are going to be confirmed by LCMS or by ELISA. We've got other ones that worked in one batch of a formulation, but then it didn't work in the next batch. So reproducibility in these nanoparticle formulations is a really big deal. But overall, it gave some information then that is useful to the guys working in Work Package 5, and which are perhaps the more interesting nanocarriers in the pipeline. Okay, we also work on permeation enhancers. So we got some data that we've already put into the public domain using sucrose laurate as an enhancer with insulin, and we were able to get pretty good insulin delivery in these jejunal or colonic loops in the presence of either sucrose laurate or C10. So this is some of the newer data we've got with permeation enhancers, where we hadn't had that data before with these types of molecules. Okay, so London, they have very good fluorescent data in mice. So this is an example of one of the prototypes selected, where they're able to show using labeling of blood vessels with this dilite 488 lectin, and also other imaging techniques. They can show that some of these nanoparticles do cross the epithelium and can be located below and in epithelia. It's not quantitative, but at least it's a clue that some of these carriers do cross, and that's in a mouse model following oral gavage. Ecam Research and Verona, they've also done a lot of work on fluorescently labeled prototypes, and they've been giving these into mice, and they've been checking for co-localization. And again, they're showing that a lot of the particles, they do get stuck in mucus, but also some of them move down the side of the villi, and they also can cross the epithelium. So that's very useful data. And Hanover, they have done a lot of bioimaging using bioluminescence with fluorescently labeled prototypes using IVIS imaging and so on. And in one prototype, or 2 prototypes that they tested using this type of in vivo imaging, they were able to show that a lot of the prototypes ended up in the stomach and in other organs when they were given by gavage into mice. And then they could pull out all the different organs and show if there's a high fluorescence signal of these particles in the different organs. So again, that was very useful information. And then finally, Ecam Research and Verona. So Verona's, one of their main assets is that they're good at working out mucosal immunization techniques, and they're able to look at expression of CD68 and CD3 on various cells within the epithelium of the intestine, and they can show whether these signals go up or whether they go down in response to various nanocarriers. So overall, we found that a lot of our labeled nanoparticles in vivo, they do attach to mucus. Some make it to the membrane, and some do get across, but it's not very quantitative. so far in that regard. But clearly, some do better than others. That's what we can tell you today. Most of the data we have in these models is qualitative rather than quantitative. The installation model was actually quite successful because it's quite simple and it's a good screening tool to show that we can deliver peptide and get a pharmacological effect from some of the carriers. And then finally, I think for the first time, some local immune events have been shown around these carriers. And the overall message is that they're not really immunologically very powerful. And that makes sense because we deliberately selected nanocarriers made from well-known excipients and agents that have been used in humans before. So that's work package 4.
Thank you very much, David. So we now have time for questions.
I was very curious about this biodistribution results. The main part of the particles were found in stomach, but the second organ was skin. And I was very curious about
Well, so are we. That's a result from the Hanover group. I don't know whether they're here today. They'll be here later on, and they will be describing that result in more detail in the consortium. But clearly, it sort of makes sense that a lot will end up in the stomach. But where they go beyond that, I think is open to interpretation.
And it could be the label also.
Yeah, the label could split off from the carrier, so this will be an ongoing QA problem for us.
Yeah.
Justin.
Is the volume of the gavage standardized amongst the different groups?
Yeah, it's mainly about 0.2 mils going into the mice and maybe 0.25 going into the rats. We didn't play with osmolarity like you've done, but I think that's an important thing that you said this morning about hypoosmolar solutions.
Another question? Oh, okay. So if there's no other questions, thank you, David. So we move to